Good afternoon, everybody, and it's really a great pleasure for me to be here. And I know many of your faces from my previous life. And I will be giving you an overview of where we stand with the microbiome field today after almost a decade of research in highlighting how it may impact many of the diseases that you are involved with. I'm sure most of you have heard about the gut microbiome, the most important microbiome in the human body. There are other microbiomes in our skin, genitourinary tract, and so on and so forth. But the gut microbiome is among the most populated microbial cultures on Earth, and it is present within each and every one of you. And they say that one picture is worth 1,000 words, and I think this scanning electron microscopy, 160,000-fold magnification from a normal terminal ileum is one of these pictures, and you can see the villi, those bumps that you meet almost on a daily basis, but you can also see the carpet of what looks like a shoelace over them, and this is the microbiome consisting of trillions of bacteria of thousands of different families, but also hundreds of types of viruses, fungi, parasites, which form this world within each and every one of us, from the moment we are born until the moment we die. And in the last few years, as I will try to show you, many of us in the field have shown the microbiome to be important in many different processes. So we are all born sterile, and we acquire our microbiome from our parents and then from our environment. And in humans, around the age of 2 to 3 years of life is the time when we acquire our adult configuration of a normal, healthy gut microbiome, which stays relatively stable. It's very relative because the deeper we probe, the more we see that it is not really stable along our entire life until we become old. And we, in my lab, regard the microbiome, if you may, as a signaling hub, which integrates into it many factors that come from our host, from us, including the host genetics, which impact the composition and the function of the microbiome, the immune system, but also many different functions or many different factors that come from our environment. And you can see here some of these factors, our lifestyle, our diet, our hygiene, and many more factors. The microbiome takes all of these different signals, integrates them, and communicates them to the host. And we in Other believe that these very poorly understood but important communication channels between our microbe and our human side are critically important in determining our health or our propensity to develop very common diseases. And basically what my lab has been doing in the last few years is trying to decipher the molecular basis of all of these interactions. And in doing so, the microbiome field as a whole has discovered that the microbiome and its interactions with the host are involved in almost any physiological function that you can think of. And you can see here some examples. There are many, many other examples coming. So almost any function of the human machine is impacted in one way or another by the microbiome. And the million-dollar question is our attempt to try to understand the mechanism of these interactions. Now, this is a very, very young field. We are less than 10 years of age, although the fact that we have a microbiome has been known for over a century. The microbiome has been regarded for many, many years to be unculturable, and therefore nobody was able to study it. And the breakthrough came in 2001 as part of the Human Genome Project, in which very advanced ways of characterizing large chunks of genomic data were developed. And we use exactly the same technologies, which we call next-generation sequencing, that were used to decipher the human genome in trying to look for the first time into those bacteria which are so hard to culture. And using next-gen sequencing and other very advanced techniques, we are now able to start and scrape the surface and understand what the microbiome is and what the function of different microbiomes are. So, for example, you may hear of a method called 16S ribosomal DNA sequencing, which is a type of next-gen sequencing, which we use to determine which types of bacteria are present within one or another microbiome. So this is a relatively simple way of doing next-gen sequencing, but we are now actually in the middle of another mini revolution in which we are now sequencing the entire genomic content of each microbe in a microbiome. So we're talking about more than 3 million genes within each and every one of us from microbial origin. So we have 150 more bacterial genes within our body than human genes. So by that criteria, each and every one of you here in the room is 95% bacteria. And we know very little about these genes, and we're trying to study them. And lately, we've been trying to study not only the genes and what these microbiomes are doing with these genes, but actually which small molecules these microbes secrete. Okay? We call these metabolites or small molecules, and we estimate that there are tens of thousands of different small molecules within our gastrointestinal tract, produced or modified by the microbiome, which signal to the host. And these are the molecules which basically are the alphabet of this language between us and our microbes. And you can imagine that many thousands of these molecules swim into our sterile host, and they can impact organs that are far apart from where the microbes actually live. Okay, so this is kind of a brave new world that we and others are discovering along the way. So this gives us the opportunity to characterize our microbial world, our inner microbial world, for the first time. However, what we are slowly discovering is that the complexity is even greater than we once thought. So the deeper we sequence, the deeper we characterize different people with different microbiomes, we discover that each of us has a unique microbiome, okay? So when we go down to the strain level and to the functions of different microbiomes, we are all very unique in our microbiome. It's like a second genome that we all carry. And this, we believe and are starting to show, impacts our variability in clinical features of the kind that you are meeting on a daily basis. One other very important tool that we use in the field is aimed not only to characterize the microbiome, but actually to start and prove that it actually does something, to prove causality, which is maybe the biggest challenge of the field as a whole. And one of the most attractive ways which we use to do this is mediated by what we call germ-free mice. These are mice that are housed in these very special isolators, and these mice are completely sterile. They have no microbiome of their own. They're kept under very, very clean conditions. They are completely sterile. And this serves two major purposes. The first is that, whenever we compare a mouse that doesn't have a microbiome to a mouse that does have a microbiome, and the germ-free mouse has some disturbance or some disease, that is the beginning of a project, because it means that the microbiome is doing something to prevent this disease. But the more important thing that we do is trying to use this model as a modeling system for human disease or human microbiome. So we can take patients from the clinic with different microbiomes that may modify or may contribute to this disease and transfer the microbiome from these patients into these sterile mice, thereby modeling the human disease in the mouse setting. Okay, so this allows us to start and probe into mechanisms by which human bacteria may contribute to human disease. And I will show you a couple of examples in my lecture. And using these next generation sequencing techniques, metabolomics techniques, and germ-free mice, the field has really began a revolution within less than 10 years of being active. And if you open any leading journal, Nature, Cell Science, almost in every issue of this journal, you will find an interesting and potentially important association made between the microbiome and one of many different multifactorial human diseases, and I've just stated a few examples here in this cartoon. Now, the million-dollar question is, of course, how and what are the mechanisms? What I will do now is I will show you a few examples of diseases which I think are very relevant to your profession and to medical professionals as a whole, which have shaped this revolution. So I think the most well-studied disease in the context of the microbiome is obesity, one of the biggest and worst epidemics in human history. We're talking about, in the US alone, over 40% of the adult population which are overweight, over 17% of the pediatric population which are overweight. It's a huge disease, a huge disease burden. We really don't understand it, and we have no ways of controlling it as of date. And around 10 years ago, the microbiome field was reborn with a really amazing observation performed by a physician and a scientist called Jeff Gordon from WashU, who did a very simple experiment. He took mice that are genetically obese. These are mice that lack leptin, leptin-deficient mice, and compare them to regular mice. These are regular mice. These are the leptin-deficient mice. These mice have no appetite suppression mechanism, so they eat themselves until they eat all the food that they can eat until they become morbidly obese. And he took the microbiome from these obese mice and from these thin mice, and he transferred the microbiomes into these sterile germ-free mice, which were completely identical to each other. And to his utter surprise, Jeff saw that mice that received the obese microbiome gained twice as much weight within 2 weeks as compared to mice that received the the thin microbiome, meaning that perhaps the microbiome is a major driver of obesity. Now, this observation at the time was met with a lot of cynicism and criticism and jokes of the kind that Nir referred to, which actually means that it was true. I know that it is true because a few years later, the same research group repeated the same set of experiments, but this time with humans. So they took human twins, some of them identical twins, but these twins were special because one of them was obese and one of them was non-obese. And the microbiome from these discordant twins was taken, transferred into germ-free mice, and exactly like I've shown you before, the mice that received the obese human microbiome gained significantly more weight as compared to the mice that received the thin microbiome, showing you already the power of the microbiome in promoting an important human disease. So this was obesity, but to our surprise, the microbiome seems to be very important also in promoting the opposite phenotype of malnutrition. So for example, in this study, the same exact technique was used in allocating in an Eastern African nation called Malawi, which has a lot of malnutrition, twins that featured the same kind of discordant behavior. So these twins had one twin that was severely malnourished and another twin which was not malnourished. And actually, this is quite common in this region. The microbiome was taken from these twins, flown to the US, where these studies were performed, transferred into germ-free mice, and you can see the result here. You can see that mice that were transferred with the malnourished microbiome lost almost 40% of their weight after getting this malnourished microbiome as compared to mice that were transferred from an identical twin which was not malnourished. Now, this added another piece of information to our puzzle because this really dramatic phenotypic difference was only observed when the mice were fed the Malawian diet, okay? Only when the mice were fed the local diet from which this microbiome was taken, you saw this phenotype appearing, and if you would turn them into a regular mouse Western diet, this really greatly disappeared. So this already tells us something very important about the microbiome, and it is that from all of these factors that I've shown you before, which impact the microbiome, perhaps the most significant one is our diet. And this is where we started to try to contribute to the field, and as you heard before, we took it one step further. We looked into a few very commonly consumed food ingredients and trying to understand how and what are the mechanisms by which they may impact the microbiome. One of them was non-caloric artificial sweeteners of the kind that I'm sure many of you are consuming in your Diet Cokes and so on and so forth on a daily basis. And there's a huge controversy in the medical literature whether these are harmful or beneficial. We're not part of this controversy, but basically the people who say that they're safe say that they cannot do possibly anything wrong because they are not recognized by the human body per definition. So this is why they're non-caloric. Our body cannot degrade them and get energy from them, which is completely true. However, we thought that maybe our human body cannot do it, but our microbes are very well equipped to break these substances down and to promote effects through them, which was exactly what we found. And this was done both in mice and in humans. And to make a long story short, we discovered that in some humans with some microbiome configurations, the consumption of these non-caloric artificial sweeteners was associated with a very dramatic change in the microbiome, which induced in the host a tendency to develop glucose intolerance, which is a little counterintuitive because many of us take these exact compounds to prevent disturbances in our blood sugar. Now, this, of course, created a lot of healthy discussion in the general media, and I can tell you that in the last couple of years, there have been really large human prospective trials which have completely corroborated our studies, including very recent ones, showing that we need to start regarding the microbiome as an integral part of our body, and this is a prime example how it can mediate effects that were not expected beforehand. To take things even further, we discovered that our gut microbiome actually can memorize previous episodes of our obesity. So if you look at the global obesity epidemic, I was quite surprised to see that around 80% of all overweight individuals in the world actually feature this phenotype, this relapsing obesity phenotype, which I'm sure is very common to many of you. It is for me, for sure. A person gains weight for one or another reason, and then he goes on one of hundreds of diets that are always very effective in the short term. He goes back to to that person's baseline weight, but then within 12 months, over 80% of all dieting individuals will regain back their weight, and they will regain a little bit more than they were before. And from cycle to cycle, this gradual obesity develops. So this is the most common pattern of obesity in humans worldwide, and if we are starting to scrape the surface in understanding obesity, we know nothing about why this pattern develops. So we took upon this challenge. We developed a few models in mice for this relapsing obesity. You can see one of these models in which mice that were induced with relapsing obesity in repeating cycles. You can see the 3 cycles in red. This is a group of mice, feature more metabolic syndrome or more obesity than mice that are in their second cycle in green, more than the mice that are in their first cycle in orange. So this is very faithfully recapitulates the human disease. And again, to make a very long story short, we found that the gut microbiome can memorize our past obesity episodes and actually drive an exaggerated weight regain phenotype, and we've figured out the mechanism of these effects and actually featured some interventions that can erase this memory and prevent weight regain phenotype. And to take things a little bit forward than that, we looked into the human microbiome effect into, or the human microbiome effects mediated by our diet. And this made me read a lot of really boring nutritional literature, which forms the basis for everything that we do in our nutritional lives. And most of these studies were performed in the 1970s. And basically, the concept is, and we all follow it on a daily basis without even thinking about it, is that we determine our diet by one of several grading systems which give grades to food. So for example, calories is a system by which we give different numbers to different foods, and you go to your dietitian or your physician, and you're prescribed the diet. An even more widely used system is called the glycemic index. It is another grading system which gives numbers to foods based on their ability to induce glucose or blood sugar disturbances. So if you would look in your smartphone, you would see that almost any food on earth has a glycemic index, and this is the most commonly used diet scoring system used. But all of these grading systems were totally based on studies in the 1970s which looked at 10 human individuals given an identical diet. So all of these were averages based on very, very small studies. So we decided to do a very gutsy move and to repeat these studies 40 years later, but rather than looking at 10 people, we looked at 1,000 healthy individuals in Israel. And we were surprised to find that different people react very uniquely to any given food. So if you take 1,000 people like we did and you give them an identical food, you measure their blood sugar rise after they eat this food with a continuous glucose measurement, you would see that many people would spike on a given food, others would not. So this grading system that gives numbers to food has no meaning on the individualized level, because if my response to a given food is opposite than yours, then one diet cannot be good for you and for me. So this is a completely surprising finding that kind of breaks the entire paradigm of what we all follow in our daily lives. And again, to make a very long story short, we continued by taking these 1,000 individuals and collecting an unprecedented amount of personalized data from each of these individuals. We sequenced their human genome, their microbiome. We measured their blood sugar responses during a week using a continuous glucose measurement that gave us 2,500 measurements from each individual. They filled out questionnaires, blood tests, and this created a huge amount of data. And then very smart students in the Weizmann Institute took all of this data from each of these individuals and used very advanced machine learning techniques which enable us to form a machine learning algorithm for each individual, which takes all of this data and enables us to predict a person's response to any given food on Earth. Okay? So this was the method. You can see one of the results here. You can see that this is the gold standard. When you go to a dietitian or to your family physician and you ask them to prescribe your diet that would impact your weight or your blood sugar control, the diet does something, because the predictability of the diet in normalizing your glycemic response is 0.38. We've measured it. But when you use this personalized nutritional approach, which we developed based mainly on the gut microbiome, but also on other parameters, you can dramatically increase the predictability to make it much more meaningful and valuable in controlling your blood sugar levels and your weight. We've further validated this by taking another group of individuals, pre-diabetic individuals, putting them through our process and asking the algorithm for each individual to prescribe a set of healthy diet for this individual or a bad diet for that individual. And then we asked them to follow their good or bad diets for another week while we extensively measured them. And we could basically cure pre-diabetes by modifying nutrition on a personalized basis. This is all measured and quantified. And what was really cool for us is that the biggest set of contributors to our ability to predict a person's response to any given food was the gut microbiome. So we basically are able to take a sample from a person, sequence them very deeply, add some clinical data, and form an algorithm, a predictive algorithm that would tell you which food is good for you or which food is bad for you. So there's not such thing as good or bad food for everyone. It is all individualized and based on your microbiome and other clinical metadata. And just to give you a very recent example of a paper that is about to be out next week, we've followed up on this new breakthrough system, and we've performed in Tel Aviv this clinical trial in which we compared two types of bread. One is the best, most finicky, handmade, whatever bread in Tel Aviv that one can make, and we directly compared to a simple white bread of the kind you find in the supermarket, which most of you would regard as non-healthy. We gave two groups of individuals one or the other bread. We followed them extensively, sequenced them every day for their microbiome, and then kindly asked them to switch their bread and continue to measure them. And again, to make a very long story short, there's no such thing as good bread or bad bread. There's good bread for you or good bread for me, and we can actually predict which response you would have to which bread based on your microbiome, which is exactly what we did. So this kind of tells you an overview of how I see the microbiome in the context of diet and how we can use it to control the metabolic syndrome, obesity, and diabetes. But the microbiome research now takes us much further than that, and I'll give you a couple of examples. For example, if you look at liver disease, almost any liver disease which you guys are confronted with is greatly impacted by the microbiome. This makes sense because the portal circulation takes many of the products which are manufactured by the microbiome into the liver, but we're slowly starting to find the mechanisms by which the gut microbiome impacts many of these liver diseases, and this opens up avenues for intervention. It goes even further. The gut-brain axis is now gaining a lot of interest in the last year, and more diseases such as autism, Parkinson, and others in the brain, a completely sterile organ, there's no bacteria in the brain, are shown to be impacted by different mechanisms by the microbiome of different people. Just last night, there was a Nature paper published on cavernous malformation, a structural defect in the brain, in endothelial cells in the brain, which was found to be completely controlled by the gut microbiome through a set of metabolites that impact immune mechanisms, and actually both in mice and in humans, this is believed to be preventable or treatable by treating the microbiome, just to show you the scope of the revolution. We are now moving into trying to understand the differences between patients in reacting to drugs. So, as physicians, we all give different drugs to different people, and whatever you take the most effective drug that you ever gave a patient, the response rates among a big cohort of patients is always 20, 30%, and the rest don't react. We always kind of, move from one drug to the other in attempting to see who would respond to whom, but we are now starting to understand that the microbiome, this huge biochemical factory which lives within each and every one of us, determines many of these personalized effects of drugs, and there are more and more drugs that are shown to be affected by the microbiome. This morning, on my way here, there was a cell host micro paper that was just published on biological treatments in IBD that can be impacted by differences in microbiomes of different patients. So this, of course, has many therapeutic potentials. And we're trying to develop the same kind of machine learning predictors to try and impact our decision-making processes in telling which patients would benefit more or less from a given set of drugs. So this is, for me, the future of applying the microbiome research into the clinical practice. And I think one of the biggest frontiers, which are really exciting for me and for many others, relates to what we call the cancer microbiome. So you meet cancer much more than me, and we classically regard most cancers to be sterile, but with these very, very advanced, sensitive, specific next-generation sequencing techniques, we are slowly starting to see that cancers are not sterile. And actually, we have identified that many of the cancers have a cancer microbiome, very low biomass microbiome. But you can see an example here of a work not done by us, but a very good friend of mine, Ravit Strassman, also from the Weizmann Institute, which is about to come out in Science, in which his group has recognized a cancer microbiome in human pancreatic cancer. And this cancer microbiome in some patients was able to degrade the chemotherapy that was given to these patients, completely explaining why these patients became resistant to this chemotherapeutic treatment. So just to tell you that by understanding these concepts, we can have a revolutionizing effect on our patient management. Okay, and you can see here a sample from one of the cancers, and you can see immunohistochemistry demonstration of these bacteria. And, you know, the field is moving and many people are joining, and we find more and more bizarre effects of the microbiome. For example, you guys are surgeons, so you probably still believe in love and in passion, but I'm here to tell you that it's all bullshit, and it's also all the microbiome, at least in flies. So this is a really cool paper looking into fruit flies, into Drosophila, and showing that when you grow two cultures of identical Drosophila, that differ only in respect to their microbiome, and then you put them together and you measure who mates with whom, who has sex with whom, you can see that there is 95% preferences between fruit flies that carry the same microbiome to mate with each other, and only a minority of them want to mate with fruit flies that have a different microbiome. Now, these are fruit flies. They're very stupid, so they don't know that they have a microbiome. So there's something to it. We don't know the mechanism, but I think it tells you how far-reaching the microbiome effects may be, at least in flies. Now, I will end by, I think, with one of the sexiest, I think, at least in my view, the sexiest concepts presented by the microbiome. Now, with all of this hype of the microbiome, you know, we forgot a little bit the human genome, which I'm telling you is extremely important. We don't disregard it. It really makes us who we are. The problem with the human genome is that, at least currently, we are completely unable to modify it in a good way. So if we have a susceptibility gene to develop breast cancer, we are born with it, we will die with it, we have nothing to do with it. In contrast, our second genome, our microbial genome, this fused genome, which is 150-fold greater than the human genome, is very much plastic and very much amenable to change by a growing number of interventions, which make it very, very attractive as potentially a future therapeutic modality or avenue. So for example, you know, eradicating the microbiome is a very non-specific way, but if you think about it, you know, we've treated cirrhotic patients with encephalopathy by impacting their gut microbiome before we knew there was a gut microbiome. So all of the interventions with antibiotics, with lactulose and so on and so forth are targeting the microbiome of these patients, and through the microbiome are impacting their susceptibility to develop encephalopathy. But we are trying to be a little bit more elegant. Of course, there is the probiotic field, which is very, very common in your supermarket. Some people are sure that it works. Others, like me, are sure that it doesn't. But it's a huge economic it's a huge field with a lot of money invested into it. And I think the jury is still out there on whether and in which conditions probiotics work. But as you've heard before, maybe, one extreme way of changing the microbiome, the extreme form of probiotics is fecal microbiome transplantation, which has been shown to be extremely successful in Clostridium difficile hospital-acquired infections. It is well into the clinical practice also here in Israel. I think for us, it's a proof of concept to the power of the microbiome and what we can achieve when we change it, when we brute force modify it. I'm not sure that this would be the gold, the silver bullet that would be used in more chronic, complicated diseases, but it's a very promising avenue. And then there are prebiotics. So we have probiotics, now we have prebiotics. Prebiotics are ways by which we are not changing the microbes, but we're changing the foods that impact the microbes. So if we change nutrition, we change the microbiome, and through the microbiome, we may impact disease. Again, this is a very problematic kind of hypey field with little reproducibility, but what I've shown you before in terms of personalized nutrition, which is science-based and measurement-based, is again a very advanced form of prebiotic treatment by which we personalize our nutrition as a way to intervene with our microbiome. And this, at least, may be promising as a way to intervene in diseases other than just the metabolic syndrome or glycemic control, but any disease that is impacted by the microbiome. Now, there's a new modality, which we've coined as postbiotics. So we have probiotics, prebiotics, and now we've coined postbiotics. Postbiotics is a way by which we understand what different microbiomes are producing in terms of the small molecules, the metabolites that they produce, and how they may impact disease. And if we find that, for example, a particular metabolite is missing in context of a different disease, of a specific disease, we can replenish the metabolite or block the metabolite if it causes in excess susceptibility to this disease. And this is very attractive to the field, and I think to science as a whole, because as I've told you at the beginning of my talk, the more we study the microbiome, the more we are certain that each of us has a unique microbial composition that makes our microbiome. So intervening through bacteria would be a little bit problematic given the uniqueness of each of us. However, understanding the common mechanisms which are mediated by the downstream metabolites would be more plausible as a more generalized treatment, and this is what we and others are making a big effort in understanding. Of course, this necessitates a biochemical, molecular level of understanding of what different microbiomes are secreting and how this impacts the human host. But if you ask me what the grander vision is, I'm pretty certain that we will end up by combining these different approaches, combining personalized probiotics, personalized nutrition, and postbiotic therapy in adding another facet of treatment to complicated human diseases. So this is kind of my vision of the field. We are all trying to characterize different microbiomes and understand variability between human individuals. We are not afraid of variability. We think that we can explain it, and this could lead us to understanding why different patients in different disease settings will have different consequences. We try to link the variability to disease susceptibility and to exploit the mechanisms which we slowly discover in constructing new out-of-the-box rational treatment mediated through the microbiome in tackling common diseases. And I will finish by thanking the very, very many people in my group that have joined me in this journey in trying to understand this really great and complicated new world. These people come from Israel, but they also come from 4 different continents, many different countries, and they're really a great bunch to work with. I will also thank my many collaborators in Israel and across the world in doing all of these projects, our funding agencies that are making our work possible. And with that, I think I'll stop here. Thanks.